So hello, everyone. Thank you for joining our first webinar in our CTMC 2023 webinar series. Today, we have our course directors on, and Dr. Will Meurer and Dr. Chris Coffey will be presenting Designing Meaningful Early Phase Clinical Trials. So I will go ahead and hand it over to Dr. Meurer, who will kick us off.
Great. So this is a derivative slide set from some presentations that we've given in past versions of the course, but also at the American Academy of Neurology to try to get at one of the first questions you have to be thinking about when you're designing a research study in terms of what outcome measures to use. So I want to acknowledge the funding for our course that has come via these two grants and the American Academy of Neurology, and also Roger Lewis, one of our course directors, who created a first draft of one of this from a prior presentation. Sorry, those are my disclosures. I get some money from NINDS. I do get some money from Berry Consultants, which does contract clinical trial design work for a variety of commercial entities. This is our website, neurotrials.training. We have past webinars, recorded lectures, and other materials there. There's also a prior outcomes webinar given by Drs. Jeremy Scheffner and Dr. Harold Adams that is really good at this YouTube link, and these slides will be made available. So sort of the main four types of things that I want to cover in this preliminary part of the talk, and then I'll hand things over to Chris, and then we'll have a discussion, is thinking about the continuum of research activities, the concept of endpoints versus outcomes, process versus an outcome, and characteristics of outcomes. What are some things that make outcomes be outcomes? So one thing to think about and when we're designing clinical trials, it's very important to put things into the proper sort of continuum of research activities. Efficacy research is classical clinical trials, often disease-specific, disease-centered endpoints, like finding a new antihypertensive or a new diabetes drug or a new drug for acute ischemic stroke, and that's efficacy research. Most of the studies that are in the clinical trials methodology course are somewhere along the continuum of proving the efficacy of a new intervention for disease. Effectiveness research is basically how efficacious treatments can perform in a real healthcare setting. Sometimes this is through stepped wedge or cluster randomized designs, and often has patient-centered outcomes that perhaps cross disease states, and sometimes economic outcomes is the bang for the buck worth it. So translational research, in contrast, is looking at this sort of implementation research, which is also along that, feels a little bit more like that last category of effectiveness research. But how do you make, if you know something works, how do you get it to enough people to actually make it work in clinical practice? Because just publishing the paper isn't really enough. This is another form of research in terms of clinical research that can be quite important. Then there's studies that are many of the studies in the implementation space are sort of hybrid implementation studies. Then there's sort of pure implementation, which is focusing on improvement of practice with little or no assessment of patient outcomes. Like if you were doing stroke research and you wanted to reduce the door to thrombolytic time, you know that leaving the brain ischemic for longer is generally not a good thing. So then there's a benefit of that sort of study that is presumed as part of the underlying biology and other clinical data from prior data sets. So motivation for choosing what outcome measure is going to be measured, or what kind of outcome is going to be measured as part of the study. And it really needs to map to the type and purpose of the research study. What is the overall goal? What are you trying to learn? What do you need to know after the study that you didn't know before? Endpoints versus outcomes. These terms are sometimes used interchangeably, and I won't necessarily say that my definitions here are the be-all, end-all on this. Lots of textbooks have coverage of this, editorials, etc. But clinical trials typically have endpoints. If it's an antibiotic trial of urinary tract infection, sterile cultures can be an endpoint, like you've sterilized the urine. If it's an analgesic trial, reducing pain by visual analog score can be an endpoint. Health services research often has outcomes, whether it's a change in configuration of the healthcare system, quality of life, satisfaction with care, total cost of care. So there's overlap between these things, and it's not always a clean distinction. But some other factors or features that are more in line with endpoints, usually endpoints are disease specific and assessed by specialized personnel, modified Rankin and stroke. Outcomes are generally patient-centered, often factorial, and may be assessed by the patient or family, like patient-reported outcomes, like a quality of life type scale. And outcomes research in general is the study of the end results of health services that takes patients' experiences, preferences, and values into account. So again, as I noted, this is somewhat of an artificial distinction, but things like blood pressure, clinical parameters, disease status, functional status, things that are MD assessed, the ALS specific scales, NIH stroke scale potentially, although that's often used as a baseline predictor and not a final outcome. And then, the more and more things are sort of assessed at the patient level and cross diseases, the more and more that they look more like outcomes. But again, it's a somewhat artificial distinction, but it is sometimes helpful to think about whether you're working with an endpoint versus an outcome. So there are process outcomes, and again, these are healthcare provider centered. So did you give antibiotics within 60 minutes in a child with suspected meningitis? And then, that is a process measure. But the outcome measure would be, is that kid functioning well in school at age 5, because of what you did in the process up front. Process is generally not an outcome. Process is easier to measure. Process often appeals to the physician-centered culture of academic medicine. It is only important to the patient to the degree that it affects the outcome, including satisfaction. So in terms of stroke quality improvement, and sorry to use so many stroke examples, but I work on our stroke team and have done stuff with improving our care processes. Reducing door to CT scan time for patients who are clearly not candidates for thrombolytics or any reperfusion is a process outcome that's probably not going to improve patient outcomes very much, unless it's like sort of helping you find hemorrhagic strokes that maybe require different blood pressure targets and so forth. But improving the process and getting them the definitive treatment faster should affect patient outcomes. So sometimes that's important to have that distinction. So I've sort of covered this. Length of stay sometimes is a process outcome that people look at, and again, that is often a really rough type of process measure because if you're sicker, sometimes you're going to be in the hospital longer, or if you die very quickly, you're going to be in the hospital shorter. So it doesn't have a very natural mapping to the patient experience. But some of them can be important. If your institution isn't giving enough patients antithrombotics, then maybe they're going to have more recurrent stroke. There is, the number of, sometimes there's usage measures that are things that happen downstream. Sometimes, as my primary specialty is emergency medicine, and sometimes visiting me is determined to be a usage measure that is sort of adverse, right? We want to get people to have better control of their disease, whether it's diabetes or Parkinson's disease, and we want them to use the emergency department less because that can reduce costs. But you can reduce ED visits by making it harder to get to the ED. You can have a lot of people in your waiting room, so people just walk away from it when they show up there. So it can potentially be a dirty usage measure, so you do have to think about it cautiously when you're using it. And sometimes if a patient has something very simple, like they have an ear, they had a stroke, but they have an earache, and it's hard to get into their primary care doctor, they can actually promptly have their otitis media treated in an emergency department. When you think about if they need to stay and work, and their primary care physician is open, there's an opportunity cost for them to actually take a day off work to have somebody look in their ear and give them amoxicillin. So it's not always a clean measure. And we sort of talked about why length of stay can sometimes be problematic as a usage measure. Quality of care measures are other options. Again, they're often based on process measures, but they may be based on assumptions between the process and the outcome. In sepsis in the emergency department, there are sort of quality of care can be assessed with giving a sufficient amount of fluids within a certain amount of time. The link may not hold up because if you give too many people too much fluids, some patients get volume overload. Some patients are maybe helped because they get better restoration of perfusion and less shock and less damage to their organs. But some patients may be hurt because they weren't that septic and you fill their lungs with fluid. So these can sometimes be a blunt instrument. So shifting gears from the sort of process-oriented stuff towards some of the characteristics when you're thinking about outcome measures, like what are some of the domains? Is this a disease-specific measure versus a general measure? The modified Rankin scale for stroke is a measure of post-stroke disability. Some aspects of it can be applied to specific parts of other diseases, but sometimes the mapping is very incomplete. For example, if you have a modified Rankin scale of 0 post-stroke, you are perfectly normal and have no symptoms. If you have a modified Rankin scale of 1, you do not have any deficits, but you may have some symptoms. So basically, you're effectively not disabled at all, but you may have new symptoms. So if you develop migraines after a stroke, your modified Rankin would be a 1 and not a 0. That being said, every person in the world with migraines doesn't start out at a modified Rankin of 1 because the modified Rankin scale is a measure of post-stroke disability. So it is, to a certain degree, undefined if you don't have a stroke. So the context matters. New headaches after stroke gets you from a 0 to 1. Headaches prior to the stroke, unchanged by the stroke, gets you, 0 to a 0. There can be proximate outcomes that are occurring in the more immediate sense in response to treatments versus long-term outcomes. Physiologic versus functional general blood pressure is a common physiologic measure. Some measures are accepted and validated. Sometimes they're newly developed measures and unvalidated measures. You should take great pause if you think you need to develop a new outcome measure because the development of outcome measures is hard. But you have to think about whether the outcome measures are important to patients, to parents, to the investigator. Who is the customer for this? And can they be assessed reliably and validly? Is it reproducible? Again, this can be through training, this can be through standardized assessments, et cetera. So again, using this sort of example of a disease-specific, meningitis can have a specific neurologic outcome like hearing acuity. That would be, not time to a sterile CSF culture. That would be a disease-specific outcome measure. Some that would be more general, like what is their performance in school? What's the Vineland behavioral score? How are they doing with activities of daily living and health-related quality of life if we're thinking about an outcome after childhood meningitis. In terms of proximate outcomes, soon after the intervention or episode of acute care, these are most likely to be affected by the intervention, but sometimes they can be misleading. Like if you get high-dose epinephrine in cardiopulmonary arrest, you may bring more patients back, but more of them don't survive the hospital discharge because their brains were not, blood flow was not restored to their brain sufficiently fast in order to allow them to survive with a good neuro outcome. So sometimes proximate outcomes can be misleading. A famous example from cardiology is the studies with patients having premature or frequent premature ventricular contractions after MI, getting prophylactic lidocaine, which reduced PVCs. So the proximate outcome was positive, but ended up increasing mortality, so that the more important distal outcome was the opposite of what the proximate outcome was showing you. So in terms of physiologic measurements, these are not patient-centered outcomes, but they even FEV1 for your breathing status, but they're related to the patient experience. Some more general outcomes, like we talked about, health-related quality of life and general quality of life. Domains are areas that are evaluated by the outcome measure, like neurologic function, behavior, maybe motor function, activities of daily living. Range is for a measurement where the the measurement actually has discriminatory capability. Standardized intelligence tests, if they have a mean in the population of 100 and a standard deviation of 10, you get a sense as to a difference of 1 point may not be very meaningful. However, the Glasgow outcome scale, where patients who are severely disabled have a different score than patients who are perfectly well, 1 point on that scale has a much bigger meaning. And then, again, accepted outcome measures versus newly developed ones. So there are typical steps in developing an outcome measure. You need to define the domains that need to be measured, developing a draft instrument, evaluation of its reliability and validity, some revision after the findings from that, and establishing the norms and further reliability and validity testing. And again, this can be very hard work. Harold Adams in that recording that I showed before talked a lot about the development of the NIH stroke scale, which was a very rigorous process, very important, and a lot of work. So particularly for people who are in this course or developing early stage clinical research in the early portions of their careers, should really try hard to use previously accepted and validated measures. This will help ensure that you have results that can map to other studies, and it will really help you get things published later. It will help you get further funding in the future, and so forth. In rare cases, a new outcome measure must be developed and validated before a research project can be initiated, but that's unusual. In certain disease states, you may need to move something over, and the degree to which it might be valid in a new population sometimes is a matter of debate. But in post cardiac arrest encephalopathy research, we do use the modified Rankin quite a bit, and it's not perfect, but in terms of the disability after cardiac arrest, it's generally accepted, and as a valid measure, it's been looked at by a variety of bodies. The major sort of deficit in the cardiac arrest research is that people with ischemic stroke, in contrast, have a lot of focal motor deficits that sometimes are contributing to the disability, whereas when people are survivors of cardiac arrest, there's more problems with executive functioning and not necessarily overt aphasia or motor weakness. But downstream, whether or not they can care for themselves, get back to work, et cetera, those things track somewhat similarly. So it's it's not perfect, but it's pretty good. So what is the importance of the outcome? Is it important to patients, parents, clinicians, insurers, or society? Who is the audience? Who's the consumer of this research, of this finding? And these should be aligned. The outcome measure should have importance to all of the constituents. And also thinking about these measurements, you got to think about reliability and validity. Whether you're going to get the same answer if you do it multiple times across multiple raters, reliability, or is it valid? Like, does if there's an accepted gold standard, which sometimes there isn't, but if you're measuring blood pressure with a new cuff, are you getting values that are close to a very expensive and well-validated cuff? All right. Some of the questions that you have to think about when you're designing research are you using a population that is similar to where that outcome measure was previously developed? Is it a similar age distribution? Is the severity of illness similar? Is the level of disability similar? So that's a key question when you're thinking about, have I got the right outcome measure? Sometimes there's sort of shortcuts, right? If you're in a disease that there's a lot of research in, you can kind of look at, look through your disease areas, studies that have been published in the New England Journal, and be like, I wonder what outcome in clinical trials, I wonder what they use for the primary endpoint / outcome in those studies. That can be a good place to sometimes look to get some hints as to what may be important. But who was administering the outcome measure? Was that similar to, are you in your study, if you're going to have caregivers administer a modified Rankin, that would be very different from what's been done in other studies in that space where a trained examiner is generally asking the questions. Is the outcome measure going to classify well? Is it going to find people, if the goal is to find people with pre-existing disability, is the outcome measure good at doing that? If that's if it's a dichotomous outcome of disabled versus non-disabled, is it finding those who are disabled? And is it noisy? Is it going to find a bunch of people who aren't disabled and misclassify them if this is a dichotomous outcome? Is there measurement bias that when you go to assess this outcome, there's going to be, patient, you only have clinic on Friday nights, and by the time everybody gets to clinic, they're really tired. So is there something that's externally pushing on the way that you're measuring things that's going to lead to a sort of systemic change in what measurement you're getting? And is it precise? Does it have this intrinsic reliability in that it's giving you basically important gradations? Can different people doing it who are adequately trained arrive at the same answer? And that is important for its validity as well. So this is I think the final slide. Is the outcome measure valid? Does it provide a characteristic? Is it providing a measurement of the characteristic you want to measure? And again, in this setting, is there an appropriate gold standard against which this validity has been tested? And if there isn't, what's the work that has gone to establish the internal validity of the outcome measure? So with that, I will say choosing endpoints and outcome measures is critical. Careful understanding of how valid they are and what the strengths and weaknesses of the validity and performance is needed as you think about this. And being able to understand the rationale and performance of the outcome measures is often a clinical issue. So again, one of the foundations of our course is collaboration between biostatisticians and clinician investigators. And sometimes, these are questions like, how much can this post-spinal cord injury outcome measure vary across the population? Or what's the trend of that over time? Even though that sounds like a very statistics-y question, it's a clinical question, right? It's like, what are these patients like when you see them in clinic? What is the expected amount of variability between two different people with spinal cord injury and so forth? Those are measurements that have statistics associated with them, but those are things that you and the statisticians have to collaborate on because you're the content expert. So that's the output of other studies. So with that, I will just sort of poll the audience and see if anybody has any questions they want to ask before I turn things over to Dr. Coffey. I think Lori didn't think I would get through 37 slides in 20 minutes, but I did. Cool. Well, I think, after Chris's talk, you may have some questions, but we'll be able to come back to this. And I will be we'll both be here to answer questions. And if you put things into the chat that you think of while Chris is talking, I will work to answer them. So thank you.
Thanks, Will. So I'm going to give more of the statistician perspective and some aspects of similar to what Will described, and slightly different in terms of how you put this together, with your endpoint and the ways that Will chose to come up with the question, and how do you kind of design it to answer your clinical question with statistical tools. Before I do that, I will make one comment on something that Will mentioned that we've actually started to encounter a lot within NeuralNext, where I work, with respect to validating endpoints. Will mentioned using a valid endpoint in another area, but even within the disease, like if you're doing a study and you're using an endpoint that was validated for the disease, but you decide you don't want to use all the questions, use a subset of questions, it's no longer a validated endpoint. Or much more commonly, what's coming up is if you take a questionnaire, 20 questionnaire, assessment that was validated in English, you have it transcribed to Spanish because you want a Spanish version of it. It technically needs to be validated in Spanish. You can't assume the validation in English carries directly over to Spanish. So those are some things to keep in mind that pop up, especially as a lot of the discussion on having more diverse clinical trial populations come in. That seems to be popping up more and more in recent days. But I'm going to talk more about kind of design. Next slide, Courtney. And in general, a lot of this hinges on what, go back up one, on what Will did. And really all of the choice of endpoint and the manner that Will talked about gets back to kind of a step further, which is what is the objective of your trial? Because your endpoint is going to be determined, by what the objective is. And any trial that you design should have one or more primary objectives where, in essence, the goal of the trial is to answer that question, right? In the affirmative or the negative. Could be, does the drug work? Is there some other aspect? Can I choose a dose? But in general, you'll have one or more main questions, and the success of the trial is, can you answer that question? A trial that shows that a treatment doesn't work is not a negative trial. That's a term that is often used. The only negative clinical trial is one that does not answer the question of interest. If you answer the question either to verify that this is a productive line of reasoning of research that should move forward, or it's not, that is a successful trial. So setting the objective is something that leads to the question, and this is very much as much art as science when it comes to designing clinical trial, in that you typically need both the clinician and the statistician working together in order to come up with what is the clinical question that I want to address, but also how can I put that in a manner that it can be quantitated and assessed and tested in some way to know whether we have answered the question one way or the other. The way I often describe this is it's very much like two individuals who speak a different language trying to come together. The clinician speaks more of the medical, jargon in terms of how to put the question together, whereas the statistician is thinking of this more from a statistical terms. And those have to meet in the middle in order to make this work. And that process is much harder, I think, than a lot of people think it should be coming in. And after you get experience doing this, I think you'll appreciate that perhaps a bit more. Next slide. So part of this is like, what is the question? And the question is not just, okay, I think drug A is well for this to treat patients with this disease, so I want to do a trial to say, is drug A better than placebo? Right? That's not necessarily a good question to know how to design the trial. What you really need is more specifics. What population, as Will mentioned, in population W, is drug A at daily dose X more efficacious in reducing Z over a period of time T than placebo? So better could mean a variety of different things. Better with respect to what endpoint, as Will mentioned, and that also needs to define to what type of value on that endpoint would be better. What is a meaningful change? And all of this drives your choice of sample size, your choice of justification for the trial, and so forth. So a lot of that will come in, and the very early discussions will hinge on these types of kind of taking a broad question and putting more specifics to it. Next slide. The primary endpoint is you choose with a lot of the properties that Will mentioned, and some of these just repeat things that Will did, but it really needs to be linked to the objective. And so the choice of the endpoint will help you to better address the objective that you hope to set out with the individual study. And sometimes there's back and forth as you flesh this out to make sure, as you're choosing your endpoint and looking at the properties that Will laid out, that you're not deviating from what you really want to do, that you're actually measuring what you need to measure to address the objective that you set out to address with the trial. Next slide. This gives some examples of, and each of these is an example that I've actually had experience with, and these are summarized in a textbook that was published from some of the people who organized a prior iteration of the course that was held in Vail and in the 2000s. But it kind of shows how, you have a concept, you have an outcome, and you have an endpoint. And so you might say, I want to do a study in order to decrease pain intensity, right? But then you have to take that and quantify that into an endpoint that you can measure and assess and test in some way. So maybe that becomes change in pain intensity as measured by a 10 cm visual analog scale over 90 days. Or for stroke prevention, decrease in stroke rate, right? Reduction in proportion of patients with stroke over a 3-year period. So you have to give some more specifics to know what you want to measure. And then that will, again, will drive the way that you design and set up your trial. Next slide. Now, there are often lots of secondary objectives, especially in more of the early phase trials like you're going to be looking at. Secondary objectives can be really informative, but they can also be somewhat problematic in that often, by definition, these are not powered. And there can be a tendency to want to throw a lot of secondary objectives in to make sure nothing is missed. But on the other hand, that can be seen in many cases as a fishing expedition. What I mean by that is you're just casting your rod out there hoping to catch something. So it helps, right, if there are some key secondary objectives to, quantify those, but you want to, choose parsimoniously. And by that, I mean, have a small number of secondary objectives that really cover the main objectives that you think will help inform the next step if you're successful on your primary endpoint. Two broad types that typically come up is that you may want to examine the impact of your treatment on some other response variable. So for instance, in a Parkinson's disease study, you might have your primary, the MDS-UPDRS, which is a more broad function of progression in PD, but you may have some other specific, more cognitive specific endpoint that you want to look at, and you might want to assess the treatment effect on that. Or another type of secondary objective would be looking at pre-specified subgroup hypotheses that you might have if you felt those with some disease you're studying, those with more severe disease might be different from those with mild, moderate disease and how they would respond to treatment. You could put those in. But a lot of the discussion will hinge on this. One of the things to keep in mind with secondary objectives with the current regulations where you have to register your trial on clinicaltrials.gov, the more secondary objectives you put in the protocol, the more things you have to upload to clinicaltrials.gov at the end of the study. So the results for every primary and secondary endpoint have to be uploaded at the end of the trial. So if you put in 20 secondary objectives, you're going to create a lot of work for yourself at the end of the study to upload. That may be something you're willing to live with, but that also kind of drives towards trying to choose carefully, what to put in as a secondary objective and is it really meaningful. A lot of other things can be thrown in as exploratory objectives, typically are, especially in early phase trials, because there's a lot of data collected that you want to assess. And in some cases, if it's secondary or exploratory, may have minimal impact in terms of your long-term goals. Next slide. So just talk briefly about phase 1 clinical trials. Most of you are probably in more what I would call early phase 2 trials. Phase 1 clinical trials would be kind of the initial studies in humans. Mostly if it's a new drug, this would be in some ways more common in drug development and industry setting than in an academic setting where you may be looking at repurposed drugs where phase 1 trials might look a little different. But in many settings, if it's the first study in humans of a new drug, you might do a relatively small dose finding type study where the goals are kind of both to get a better sense of the PK/PD aspects of the drug, but also more critically to kind of look at how high can you go, right? What's the maximum tolerated dose? How high can you go in the dose range before bad things start to happen in healthy participants, which if bad things are happening in healthy participants, you're probably not going to be able to dose that high or higher in diseased individuals. But this is often used to try to establish a range of doses to consider once you move into later phases. With the general idea, this is not always true, but typically it's assumed that the higher the dose, the higher the risk, but also the higher the chance of efficacy. So the highest dose that you can get to that is safe should maximize your chance for efficacy. Like I said, that's not always the case, but that's in general a sense that many of these phase 1 trials try to address. Next slide. What is probably more relevant to those of you in the course with the trials that you propose would be more of a phase 2 trial. Phase 2 trials are in some ways, not in some ways, probably in every way, is that some of the most difficult to design because phase 1 you're just looking at safety. By the time you get to phase 3, it's pretty clear what the question is. I mean, they're not easy to design, but it's pretty clear what your question is, and you're looking at more confirming whether it works or not. In phase 2, we're in more of the proof of concept stage, where we're really trying to do two things. One, get more information about safety to verify that there are no major safety concerns moving forward, and two, that there is enough evidence of efficacy that would justify moving forward to the larger, more complex, more expensive, phase 3 confirmatory clinical trials that are in many cases going to be multi-site, large endeavors that may take a large number of participants over time. So because of that, you're trying to address efficacy in phase 2, but not powered for efficacy in the same way that you would think of in a phase 3 clinical trial. So what you really want to do here is think about, your sample size is a trade-off between lots of different things. Type 1 error we really worry about in all clinical trials, in particular, phase 3 clinical trials, but also in phase 2 clinical trials, which is the concern about saying, a treatment works when it really does not. But in a lot of phase 2 trials, you may be equally concerned with what we would call a type 2 error, not showing that a drug that actually worked does work. And so it is not uncommon in some phase 2 trials that you may use a higher type 1 error. I've seen phase 2 trials with type 1 errors of 10%, even as high as 20%, with the goal of not missing, not having a type 2 error. A lot of that affects how much risk you want to take with your go, no-go criteria in your phase 2 trial, in your phase 3, because obviously the more stringent you are in phase 2, if you meet your criteria, you should have, in theory, a higher chance of success in phase 3. The more lax you are in phase 2, the more risk you're taking in terms of your phase 3 trial. But these are all things that need to be kept in mind in terms of doing this. It is argued that phase 2 trials, even though they're larger than phase 1, they're smaller than phase 3, and there is an argument that they're smaller than they should be. But part of that is a tension between trying to get as much information as possible before going to phase 3 to make sure that you've addressed all the key questions you need to know to go to phase 3, versus trying to get to phase 3 as quickly as possible, because if the drug works, you want to be able to show that it works as quickly as possible. So there's many ways a natural tension there between, if you spend a ton of time in phase 2, by the time you get to phase 3, you'll have a lot of questions answered, but it's going to take you longer to get to phase 3. And that is something to keep in mind as well when looking at some of the trade-offs. Next slide. So as I mentioned, kind of the main goal is to provide an assessment of preliminary efficacy, screen out ineffective treatments, determine if a new treatment is sufficiently promising to justify going to a large-scale randomized clinical trial. And in many cases, also another aspect of that is to evaluate potential study endpoints and make sure that your endpoint is the right one to use by the time you go into your confirmatory study. It's also important to characterize the safety profile. One of the things that I will mention there is whether it's phase 1, phase 2, one of your objectives should never be to show that the drug is safe. You will never show that a drug is safe in a clinical trial. As you progress through larger and larger clinical trials, you can rule out larger degrees of a drug being unsafe, but you can never really show that a drug is safe. Even after approval, you can't necessarily say that a drug has been shown to be safe, because once millions and millions of people start to use it, you might see rare side effects that didn't come up in clinical trials that appear. But what we're really looking at in phase 2 is there some major risk that would either A, lead to not wanting to move forward, even if it looked like there was efficacy because the risk is too high, or B, is sufficient enough that there should be aspects of the design of your future trials that would try to minimize the risk of that particular adverse event. The most critical thing that I can say, particularly if you're submitting a grant to NIH, is to, on all accounts, avoid an early phase design that really looks to be just an underpowered version of the phase 3 study. So if you're designed and you're doing an efficacy test that needs 1,000 participants to do that, and you plan to do a study with 200 individuals, and you're going to run that test, and you're just hoping to get estimates that will help you power the study, that is not going to fly through an NIH study section. And also the estimates you get are going to have lots of variability. But more importantly, if that's your main goal, you're not answering a question in this study. And there's really, in some sense, no way that the trial can fail because you're just going to get estimates. You've already decided what you want to do. So you either have justification and you should just go straight to the phase 3 trial, or you're doing the wrong phase 2 trial because you're not really informing anything that you need to inform. One of the kind of perceptual way, and this is very difficult, I think, from the investigator side, because we talk about a concept in clinical trials called clinical equipoise. And from an ethical perspective, you should really never, ethically, you shouldn't be doing a clinical trial if there's not some degree of equipoise, where there is uncertainty in the field, whether what you're studying truly works or truly doesn't work, or has a promising risk-benefit ratio versus does not. And if you think about that from kind of an ethical standpoint, it's not considered ethical to randomize a participant into a study if you know what the best treatment should be. If there's a degree of uncertainty, then it can be justified. We really don't know. The idea of the study is to justify that. When I talk about this in our clinical trials course that I teach at Iowa, I always talk about the grandmother test. I mean, you could make this the mother test, the brother, the sister test. But in my mind, from a researcher's perspective, if you would not, if your grandmother, mother, brother, sister had the disease of interest, if you would not recommend that they enter the trial because you would be uncomfortable with them getting one of the two options, you shouldn't be enrolling somebody else's grandmother, mother, brother, or sister into the trial. So it's important to kind of keep that in mind with respect to how we're putting all of this together and trying to assess the endpoints and maintain the integrity of the study. Next slide. Now, one of the concerns about this is that, oh, sorry, let me go back. So can you go back one? The point I actually forgot to make there was we often think about as investigators that we hope to show that the treatment works in the phase 2 clinical trials, because even though there's this concept of equipoise, no investigator who's leading a trial has equipoise. They're convinced that it works or they would not be doing this. We're talking more about a societal level. But in all honesty, a very good phase 2 trial should be powered to identify treatments that are not promising as much as that are promising. And we all know that there are more clinical trials. This is especially a problem in neurology, more clinical trials that will fail to show that a treatment is effective than will show a treatment is effective. Because of that, if all of our designs are set up with high probability to show treatments are effective, whether or not they are, that's going to be problematic, and we're going to be taking things forward that we should not. So one of the key questions to ask when you're designing this, can the trial fail? And if the trial can't fail, it's probably, by fail, I mean if the treatment doesn't work, will you still show that it works in your trial? If that's the case, then it's probably a poor design and it's not really addressing the question that you originally set out to address. Next slide. So one of the things, when you talk about this and this gets into this whole go, no-go criteria, where there should be some criteria that would specify if you meet this criteria, it would justify moving forward. If you don't, it would not. And then you can, through various tools, calculation, simulation, however, working with your statistical collaborators, come up with a sample size and a design and a set of rules that would have, in theory, high probability of moving forward if your treatment works in the way that you specified, and low probability of moving forward if it does not. And so you would want to address that. Now, there's a tendency sometimes to want to cast a wide net. For instance, if you had, say, 4 endpoints, you weren't clear which is your best endpoint, and you said, I want to design a trial, I'm going to look at all 4 endpoints, and if either endpoint shows a positive trend, not worrying about statistical significance, we're going to go forward to phase 3. It's not difficult to run a simulation or do calculations that would show if that's your rule, even if your treatment doesn't work, you're going to move forward with high probability. And so that would be a case that would fall into what I mentioned before. It's a trial that really can't fail. You're most likely going to go forward whether your drug works, works at the level you want it to work, works somewhat, but not at the high level, or doesn't work at all. It's not necessarily going to change your answer. And if that's the case, it's probably not the best design, because you're spending a lot of time and resources and not answering the question that you originally set out to address when you sat down and started to design the trial. Next slide. I put on here just a list of different types of phase 2 clinical trial designs. A lot of discussion in your small groups may get into one or more of these, depending upon your specific project. These fall kind of in 2 broad types. There are those that use different endpoints than a phase 3 endpoint. So you might have a phase 2 that's more focused on a safety tolerability design, or one that's using a surrogate endpoint. It would need to be a fairly well validated surrogate, but if you have a surrogate that can be measured in a shorter period of time, your phase 2 could screen out on the surrogate with the idea that if the surrogate is well correlated and well validated with your clinical endpoint, that if you meet criteria on a smaller phase 2 trial looking at the surrogate, you would have high probability of having success on your clinical endpoint were you to conduct a phase 3 clinical trial. The other broad type of phase 3 endpoints are really those that kind of address this concern about underpowered phase 3 trials, but would be trials that you could do when you really can't use a different endpoint than in many cases the same endpoint you would use in the phase 3. So there are other designs, two-stage designs, predictive probability designs, N-of-1 designs in some cases with rare diseases, selection designs, utility designs that can be used to have some rigor and criteria in phase 2 that needs to be met, that are not powered, they're not as large as your phase 3 efficacy trials, and can address this screening in a way that has the validity in the sense of doing what you want it to do. Next slide. Just a couple of common errors in drug development that come up. I mean, one, we've kind of hinted at this as we've gone through kind of both discussions, is poorly designed, optimistically interpreted proof of concept studies. If you had no go, no go, and you're just going to collect the data and look at it at the end and make a decision whether to go, you can show two investigators the exact same results, and one who is convinced the drug works will see it in a positive light, and one who is convinced the drug doesn't work will see it in a negative light. So having criteria, having rules specified in advance can avoid, that uncertainty with respect to justifying the choice at the end. Another common error, and this is another thing that should be considered as you move through early phases, is making sure you're adequately exploring the dose, the regimen, the route of administration. And some of this comes to the other thing. And when you look at can your trial fail, part of the other question you should ask is, if your trial fails, other than the drug not working, why do you think it might have failed, right? If you said, if your trial failed now, do you have any reasons why? And you say, well, I wasn't completely sure about the dose. Maybe I chose the wrong dose. Then perhaps in one of your early phase studies, you should be exploring dose ranges to see whether, you might be able to use a different dose before you launch into a larger phase 3 trial. The other is this idea of wanting to get quickly to phase 3 because you're convinced the drug works, really without adequate early phase data. And so there is a lot to gain by a lot of the early phase studies. One of the common differences, which is somewhat unfortunate in NIH funded trials or conducting clinical trials in the NIH funding framework, is we tend to think of trials in more of a one-off scenario. We're going to do this trial and then we'll figure out what the next plan is, which is very different from the industry drug development approach, where each trial is part of a larger drug development program, and it's more clear where each trial fits into the larger framework and how they connect together. I think trying to think of that as you're writing a grant, not necessarily in the full range of a program like industry would have, but in the sense of, if this trial works, right, I meet my go criteria, what is the next step? What would my next study look like? And that can help you both in designing your trial, but also, in all honesty, in writing your grant application, because it answers from a grant reviewer, it answers a very important question, the so what question, right? If you do this and it works, so what, right? What does that tell me? What does that mean moving forward? Next slide. I just come back to this just to reiterate, there are three basic requirements that would be true for any clinical trial. It should examine an important research question. That's first and foremost. It needs to maintain and be based on a high level of scientific rigor, and it needs to be based on ethical considerations and be designed in a way that the risks to subjects are minimized. Right? Due to limitations, especially in the phase 2, where you don't have as large sample size, addressing an important question can be more of a challenge, but this really magnifies the importance of study planning. It's really critical to think about a lot of the issues. It is extremely rare that from the time you think of a question in a clinical trial to when you have a pretty good skeleton of your trial design would be less than somewhere from 3 to 6 months on the best case scenario. It's going to take a long time to make sure you're putting this together. So coming in like July and meeting with a collaborator and say, I want to submit this clinical trial in October, let's flesh this out and do that. It's not impossible, but it's going to be pretty unlikely unless you're meeting, a couple hours every day, because each conversation is like one step forward, 2 steps back until you eventually iterate on the correct conclusion. So next slide, there are some links that we put in here. These slides will be posted, which have some other information that are relevant to what both Will and I have talked about. And so at this point, I think we'll open it up for questions.
Chris, that's great. One thing that resonated about some of those last points, when people are writing grants for the NIH, there's the grading criteria or the scoring criteria of significance and approach that are often the most highly weighted criteria. The idea of significance is, it can't be emphasized enough. It is, will accomplishing these aims move the field forward, right? Like, are you learning something that's important? And did you provide enough evidence that this is the right thing to learn next? It's not like stroke is a bad disease. Everybody knows stroke is a bad disease. It is, what is the value of the research question you are asking towards moving the field forward? Because certainly some, less common diseases than stroke, you can be asking highly significant questions within those fields that could be transformative for those disease states. And they're not, any less significant because the disease affects fewer people. So just, even though sometimes words in medicine and science have slightly different meanings, in terms of NIH grants, significance is really couched towards the value of the information. And I think in this case, when you're designing the clinical trial, what is it that you're going to know after it that you didn't know before? And I think that's very important. But seems like, Chris, we were so clear that everybody's like,
I'm sure that was it.
Drop the mic.
Looks there's a couple in the Q&A.
You guys have a couple of questions. So
do you want to read them, Lori?
I can read them, yeah. Could you go into the types of trial designs using the phase 3 endpoints a bit more? 2-stage designs, predictive probability designs, N of 1 designs, etc. And the next question is an easier one to answer. Is it appropriate to use the term pilot trial? Is it considered synonymous with phase 1 or phase 2?
I'll start with the second one. And you can use the term pilot trial. I would recommend that you probably don't. Pilot trial has kind of become synonymous with underpowered phase 3 trial. And in all honesty, it's not. We talk about it in the more standard language as phase 1, phase 2, phase 3. But some of the other language that is used, which I actually think is more reflective of what you're doing, is learning stage clinical trials, which would be a combination of what we typically think of as phase 1 and phase 2, and confirmatory clinical trials, which is more of what we think about as phase 3. So, if you called it a pilot trial, there's nothing wrong with it. But I would probably suggest trying to not use that term and use more like, learning phase or early phase, or even, phase 1, phase 2.
And there's a statement out there about the design of pilot and feasibility trials. One of the lectures that Chris references that's available on the website, Lahana Thabane's lecture on pilot and feasibility studies. I would echo Chris's comments. Sometimes, though, it may be a feasibility study, but I will just sort of say that if all you are addressing is feasibility, that's often not exciting enough for funding and other things. Like, you really need to be answering some sort of exploratory phase, whether it's proof of concept, is this drug doing what you think? There usually needs to be a little bit more to it than pure feasibility. Although sometimes if there's a real question as to whether people would consent to a study, maybe you need to be doing some observational survey research first to say, like, this is what the study would like. And you show a bunch of people with the disease, like, would you enroll your kid in this? And if they're like, yes, I would, or no, I wouldn't, that gives you some feasibility data to sort of establish that doing a more later proof of concept study is the right next step.
And then for the second one, I can quickly do that. I mean, that's a whole probably hours worth of talk in and of itself. To talk about these different trial designs, and they depend on different settings. Predictive probability designs is a good one where there you're using Bayesian modeling for the most part, where you kind of say, this is the phase 3 trial I would do and roughly how big that would be. And then as you get data in your phase 2, you do a predictive probability, say, based on what I've seen here, if I were to do the larger study that would be phase 3, what would be my probability of success? And you can set that up typically where you have a lot of platform trials that are more into phase 2 actually do this, where, as you proceed through the study, if the predictive probability is higher than some specified amount, then you might graduate and go to the phase 3, where that says there's sufficient evidence that's assessed to justify moving to a phase 3 trial. If the predictive probability falls too low, below some threshold, you would stop for futility, and basically that would be your no-go. It's not worth doing. Or you reach some maximum sample size, and then your predictive probability is somewhere between the two. And at that point, it's kind of a larger discussion as to whether, where that falls, what else does it tell you, and whether that justifies the risk moving forward. There's, the futility design I mentioned, that's another one where the question you're asking is not so much, does this drug work, but is there enough evidence that it doesn't work, that it's not worth going to phase 3? It sounds like a crazy question to answer, but it's interesting that a study was done a few years ago out of MUSC where they showed that a number of stroke trials that failed in phase 3, had they had a futility design in phase 2, they might have never made it to phase 3. So there are some things that phase 2 trials, with futility design can tell. As far as the others, there's not really sufficient time to go into that with each of them, but that's kind of the broad language of where those go. And, we can get into that more in the course or within your small groups of specific types.
Yeah, and I guess I would also add that, part of it is if it's, the predictive probability designs and futility designs are sort of doing the same thing. Is there a zone of difference between, drug A and drug B that is promising enough to go to the next level, right? Like if they're exactly, if your new drug is exactly overlaid on the old drug, it's like they're both got exactly the same treatment effect, then if you have a really big study, there's still a pretty low likelihood that one's going to be the better. If it's 2% better, what's the chance that there's actually a 3% difference or a 4% difference? It may not be that much, but if you're planning to do a 50,000 patient follow-up study, that 2% difference that you observe, may be sufficient to justify that in a larger study. If something's like 5% below, it's very unlikely that it's going to beat the other treatment in the follow-up study. So that's just one sort of, I guess, simpler way of looking at those, but
Right.
You carefully have to set up those boundaries and those sort of thresholds in advance.
I think one thing before we go to the next question, from a grantsmanship side, and sometimes this is the truth, recognized, but you don't want it to come through in your grant because it really reflects poorly when it's being reviewed, is if you're mainly doing this trial, not because there might be a clear sense of what next steps are in your head, but you're doing it because you need funding for your career to move forward, and this is what you cobbled together. That might be true, but you want to think about where could this go in the future, because if it comes through as that's just what you're doing and there's no thoughts for where it would go, that's not going to be reviewed very well when it gets to there. But the significance, as Will mentioned, is not going to be that high because it's going to be clear.
So we're kind of out of time. I saw that you guys put some things into the chat. So there's the pilot and feasibility trials, YouTube, that's available. And then we'll kindly put in the implementation link for Melissa's question. Thanks everybody for your attendance today. And thank you, Will and Chris, for your, as ever, very important and sage advice to all of us.
Yeah, thanks everyone for joining.
Thank you, everybody.
Bye.